Hey, grade 11 learners, welcome to Life Sciences lesson. My name is Ntombi Dladla. This lesson is brought to you by Sci-Bono Discovery Center in collaboration with Gauteng Department of Education. Welcome, grade 11s. Today's focus or today's topic is the revision program, and we'll be focusing on paper 1 topic. And basically there are 5 topics that are tested or that are examined in paper 1 at the end of the year. However, for today, we'll be focusing on the following 3 topics, which is photosynthesis, excretion, and cellular respiration. Now, I want us to look into the overall paper of paper 1, or into the structure of paper 1. Remember that at the end of the year, you will be writing 2 papers, paper 1 and paper 2. Paper 1 will be out of 150 and paper 2 will also be out of 150. Each paper will be for 2.5 hours. However, today's focus will be dealing with the topics which are assessed in paper 1. There are 5 topics which are assessed in paper 1. The first one is photosynthesis, which carries 32 marks. Excretion, which carries 32 marks as well. Cellular respiration, which carries 22 marks. Gaseous exchange, which has 32 marks. And animal nutrition, which carries 32 marks as well. And all these marks, when we add them, they add up to 150. And remember that at the end of the year, the weightings for SBA are 40%. And most of the weightings come from the examination, which will be 60%. What does that mean? It means that you must work very, very, very hard. You must obtain much of the marks in your examinations, because your SBA only carries 40%. Now, today, as I've mentioned, that we'll only be focusing into the 3 topics, which are photosynthesis, excretion, as well as cellular respiration. Now, how do you prepare yourself for the end of the year examination? You must use the following resources: your DBE study guides, your remote learning material, the past papers from different provinces, from past years. You may also use your teacher's notes as well as the activities which were done throughout the year, so that you'll be fairly prepared for the examination. You may also refer to the exam guideline and all the relevant textbooks, all the relevant grade 11 textbooks. And do not forget our grade 11 lesson broadcasting, as well as the PowerPoint presentation that we did throughout the year. Today's focus will be on the challenging concepts for the 3 topics that I have highlighted earlier on. Now, the first topic that will be examined at the end of the year in paper 1 is photosynthesis. Now, I've started with the multiple choice question. And remember, when we were doing our broadcasting, when we were doing all the lesson broadcasting, we have learned that when we attempt or when we answer all the multiple choice questions, we must start first eliminating all the obviously wrong answers. Then you'll be left at least with 2 possible answers. And then out of the 2 possible answers, that's where you are going to choose the most correct answer. Now, let us look into this question. Here is a multiple choice. And remember, with the multiple choice question, you do not write the whole the answer, the letters or the sentence, but you provide the letter that is linked to the answer of your choice. Now, the first question says the chemical reagent that is used to test the starch or the presence of starch. Is it the bromothymol blue? Is it iodine solution? Is it lime water? Or is it methylated spirit? Remember, I said you must eliminate all the incorrect answers. Definitely, we are not using the bromothermal blue to test for starch. So the first one, we are eliminating. It is incorrect. Is iodine used? We'll check. Let us check the incorrect one. We don't use the lime water as well to test for the presence of starch. We also do not use to test the presence of starch. We do not use methylated spirit. So what does that mean? It means that the correct answer is iodine solution. But when you write your answer, you do not write the iodine solution. Which letter is linked to the iodine solution is B. It means that the correct answer for your for that question is iodine solution. Do not forget that when we test for the presence of starch, we always use iodine solution. And if the starch is present, it means that the iodine solution will, the starch or the substrate that is tested for starch will turn blue black when iodine solution is used. So if the starch is present, then the substrate that is tested for this for starch will turn blue black. Don't forget that. The second question says the process that takes place in leaves which uses carbon dioxide. Is it cellular respiration? Is it combustion? Is it photosynthesis? Or is it transpiration? So we all know that the process that uses leaves, because they are green, in the leaves there's chlorophyll, and it also uses carbon dioxide. We know that the process is what? Is photosynthesis. In other words, cellular respiration does not use those substances. Combustion does not use that. Transpiration does not use that. But the process of photosynthesis takes place in the leaves, because in the leaves, that's where chlorophyll is found. And it also uses carbon dioxide from the atmosphere. So the correct answer for that is, of course, C. Do not write photosynthesis, rather write C as the answer. 1.3, a factor that increases the rate of photosynthesis. Remember, when we did this topic of photosynthesis, we looked into the 3 factors that are increasing the rate of photosynthesis. Factors such as light intensity, factors such as temperature, factors such as carbon dioxide. So all the 3 factors, they increase the rate of photosynthesis only if they are also in the high levels. Now, which factor will increase the rate of photosynthesis? If light intensity is low, no, it does not. If temperature is low, no, it does not increase the rate of photosynthesis. If carbon dioxide concentration is low, it does not increase the rate of photosynthesis. If light intensity is high, yes, if the light intensity is high, it means that the rate of photosynthesis will also increase. So the correct answer there will be D. And then 1 point, the second question, the fourth question, 1.4. Here you are given the graph which which has the concentration of carbon dioxide and the rate of photosynthesis. Now the question says, between which points of the graph is the rate of photosynthesis increasing fastest? At which points where the rate of photosynthesis is increasing the fastest? So is it between 1 and 2? Is it between 2 and 3? Is it between 1 and 3? Is it between 3 and 4? Is it between here? Is it increasing fastest there? Is it increasing between this one, the second one, 2 and 3? Is it increasing between 1 and 3? Or is it increasing between 3 and 4? At which rate is it increasing the fastest? So you can see here, between 1 and 2, that's where there is a rapid increase. So the answer will be what? Will be A, because that's when the option is at 1, between 1 and 2. The option will be A. And then we move to the second activity. And then remember, with the column questions, you must follow the instructions, grade 11. When you are answering the column questions, please follow the instruction. The instruction says, if your answer is A, you must write A only. if the answer or your choice is B, you must say B only. If your choice is both A and B, you must say both A and B. And if the answer is not applicable in either A or B, you just say none. Don't say none A and B. If you said none, then you have covered your answer. Now the first question says, molecule that stores energy. Is it ATP? Is it ADP? Is it both of them or none of the provided options? So we know that the molecule that stores energy, can it be during photosynthesis or whatever the process where energy is stored? So the molecule that stores energy, we all know that is ATP. So the answer will be A only. For now, I am guiding you. I am also giving you the answer so that you can recall on what we did earlier on or during the course of the year. So the answer there will be your ATP. We know that ATP stores energy. The second question on the column, what stores chlorophyll? The storage of chlorophyll. Is chlorophyll stored in the grana? Is it stored in the lamella? Is it stored in both of them, or it's not stored either in the grana or in the lamella? So we know that chlorophyll is stored in both the grana and in the lamella. So what will be your answer? The answer will be both A and B. You don't encycle, you don't say A/B. If you write A/B, you are going to get it wrong, because in other words, you say it's A or it's B. But your answer should be A and B, as it is written on the instruction. Here it is both A and B. So that is how we are going to answer that question. So chlorophyll, do not forget that it is stored in the grana of the chloroplast. It is also stored in the lamella of the chloroplast. 3.3, the hydrogen carrier in plants, in light phase. The hydrogen carrier in the light phase. Is the hydrogen carried by the NAD? Is it carried by the NADP? Is it carried by both of them, or it's not carried by either of the two? So what is your answer? We know that during in the light phase, remember that light splits water into hydrogen and what? And oxygen. And then this hydrogen should be taken or should be carried by the hydrogen carrier. And we know that the hydrogen carrier in this instance will be what? NADP, to give us NADPH. So meaning that the NADP is the one that carries what? Hydrogen during the what? During the light phase. So your answer there will be B only. B only. So if maybe you are confused, you want to do your guesswork and then you say it's A or you say it's B. So remember that you always mark the first option or the first answer, even though you at the later stage you thought that B could be the answer. But when we mark the first one. And with this type of questions, we only need one option. We only need one answer. So if you guess here, believe you me, we are not going to get either of the marks. So that was very, very simple. Grade 11s, you cannot afford to lose that 6 marks. Now activity 3. Before we can do the activities, remember I said today we are going to focus in photosynthesis as being one of the topic or the concept that is assessed in paper 1. Now when you study photosynthesis, the organelle that you must always think of, or the organelle that is responsible for the process of photosynthesis to take place, is chloroplast. What does that mean now? It means that you must be able to draw and provide the correct labels for the chloroplast. This is a possible question, or this is a possible drawing that you might be asked to draw at the end of the year. Why am I saying it's a possible drawing question? It is because the structure is very simple to draw. Remember, we do not ask you to draw complex or difficult drawings, but when you look into this structure, it is very, very, very simple for you to draw. So it is one of the possible drawings or structures to be asked to draw. And remember, when you are asked to draw whatever the structure, you must be in position to do what? To provide correct labels. So do not forget to practice drawing the structure of the chloroplast and providing all the correct labels on that one. Now let us look into this question, the questions which are based on the structure. Name the organelle in this diagram. I've already gave you the answer. For the fact that we are focusing in the topic of photosynthesis, so the organelle that is responsible for photosynthesis is what? Is the chloroplast. So the answer for the organelle, the answer for number 1 is chloroplast. This organelle is chloroplast. The second question says, provide names for labels A to D. However, here we've got labels A to what? A to E. So we can safely, if we can also provide label E. Let us say provide labels A to D. In other words, you must know all the parts of what? Of the chloroplast. Now, how do we label here? Remember the chloroplast, already you are given the walls or the double wall, the double membrane, which is the outer membrane. Now you need to know what is inside now, the double membrane. The first one, A, you must give us the answer for A, which is the stroma. The B, you provide all the answers, the C, the D, as well as the E. So let us quickly label all those. So A is at the stroma. You see, that's where all the other parts of the chloroplast are embedded on. And then B is our lamella. Remember, we said even chlorophyll is found in the lamella. So B is the lamella, and then what is C? C is the starch granule. So with the starch granule, this part explains itself. Starch granule, it means that that's where starch is stored. Do not forget that. And then what is D? A D is a stack of thylakoids. And then what is the stack of thylakoids called? It is called the granum. So this is one granum, and then if there are more than one in plural, we are talking of the grana. One granum, but in plural form, we'll be talking of what? Of the grana. So that is the stack of the thylakoids. And then what is E? And then E is our thylakoid. When it is one, it is a thylakoid, And then when the thylakoids are stuck together, what are they called? They are now called the grana or the granum. What process takes place in this organelle? What process is taking place in this organelle? That is very simple. I've already explained that earlier on, that the process of photosynthesis needs the organelle, which is chlorophyll. So the process that is taking place here is photosynthesis. What is the function of C? What is C? Already we have labeled C, and then we said C is the starch granule. And I've explained that the starch granule, that part explains itself. What does that mean? It means that that's where starch is stored. So C is there, and the function of C is to store starch. So you see, these are very, very, very simple questions and very simple to answer. But you'll only be able to provide correct answers when you have prepared yourself for the paper, when you have studied, practiced, and practiced. And then the second part of photosynthesis is based on this structure or on this diagram. Remember that the process of photosynthesis takes place into two phases. The first phase is our light phase, don't forget that, and then the second phase will be the dark phase. And then light phase, that means this is the phase that is highly dependent on the light, and then the dark phase is the phase that is light independent. In other words, for the process of photosynthesis to occur during the dark phase, light is not needed. Do not forget that. Now, what is happening during light phase? The most important thing during light phase that is happening there is that this phase, as I've said earlier on, that it needs light to take place. It also needs water to do what? to take place, for the process of photosynthesis to take place. And then this light during light phase is the one that is going to split this water molecule into what? Into hydrogen and into oxygen. And don't forget the process whereby light splits water into hydrogen and oxygen is called photolysis. Photolysis is that process. Photolysis. Photo means light. So photolysis is the process that whereby light splits water into hydrogen. There is our hydrogen into hydrogen and what? And oxygen. And then where does this oxygen go? It will go to the atmosphere. And then what is going to happen with our hydrogen? The hydrogen will be taken by the what? By the hydrogen carrier. We have already explained that. We have already answered that. It will be taken by the NHDP. So it will be taken by the hydrogen carrier. So the second phase is the dark phase, and then during the dark phase, what is needed there? During the dark phase, carbon dioxide is needed. And then when carbon dioxide is used or is required during the dark phase for the process of photosynthesis to take place, what is going to be produced? Glucose is going to be produced. And then that glucose is the one that will be stored in the form of what now? In the form of starch, where in the starch granule. So I've just summarized the two phases. So now I want us to look into what? Into the questions which are posed on the diagram. The first question says, identify phases labeled as phase 1 and phase 2. Identify this phase, identify that phase. I've already explained to you that photosynthesis takes place in two phases. It takes place during the light phase. It also takes place during the dark phase. So the first phase or the phase 1 is our light phase, whereas phase 2 will be our dark phase. Now, how do you see that this is a light phase? It is because during light phase, remember there's photolysis, and photolysis broke or split down the water into what? Water into H2O, water into oxygen, and into what? Into hydrogen. So that's why we are safely to say that this is a light phase because of the hydrogen that is now produced. Second question says, provide the two raw materials labeled A and B. What are the raw materials? For the fact that you now know that phase 1 is light phase. So what are the raw materials that are needed during light phase? I've already explained that during light phase, water is needed and light is also needed, as this phase explains itself that it is a light phase. So what is needed there? Light as well as water. So A can be water, it can also be light. B can be water, or it can also be light, either way. So A is light or water, B is light or water. So these are the two raw materials that are required for the phase, for the light photosynthesis to take place during light phase. Name the byproduct labeled C. What is the byproduct labeled C? In other words, what is given off during light phase? That is very, very simple. I've said earlier on, remember what the light split down water into this hydrogen and into what? Into this byproduct. So the question wants to know, what is this byproduct called? The byproduct will therefore be oxygen. Which substance labeled D is essential for phase 2? Which substance is essential for phase 2? We now know that phase 2 is our dark phase. And earlier on, when I summarized the dark phase, I said carbon dioxide is needed for phase 2. So the substance that is essential for phase 2 here, which is labeled D, is of course what? Carbon dioxide is our carbon dioxide. So you can write it in full. You can write carbon dioxide, or you can write CO2. Either way, that is also correct. From where in the substance labeled D does carbon dioxide come from? Where does carbon dioxide come from? We all know that carbon dioxide comes from, of course, from the atmosphere. You see, grade 11s, these are very, very, very simple to do what? To answer, provided you have prepared yourself. Number 6, still on the diagram here, name the product E that is produced during phase 2. What is this product that is produced during dark phase? Don't forget that phase 2 is our dark phase. So what is the product called? Remember, during dark phase, that's where glucose is produced. So the answer is glucose. And in what form is glucose stored in the plants? In what form is glucose stored in the plant? It is stored in the form of what? In the form of starch, grade 11s. And then activity 5, here we are given an investigation. So let us quickly go through it. A tomato farmer was able to produce crops in a greenhouse which were heated to between 15 degrees Celsius to 22 degrees Celsius. In addition, carbon dioxide levels were increased from the normal 0.03 to 0.1. Now the first question says, state one way in which the farmer could have enriched the air inside the greenhouse with carbon dioxide without burning any fuel. So when you answer this question, just think of all the sources of carbon dioxide emissions. What are the sources? What are the sources that emit carbon dioxide without burning of fuel? Remember, burning of fuel releases carbon dioxide. But the examiner here, he wants you to mention all the carbon dioxide emissions that can be that can enrich air inside what? Inside the greenhouse, without, do not mention burning any fuel, because already we are giving that as one of the carbon dioxide emissions. So the answer here will be what? The investigator could have pumped what? Carbon dioxide from the tanks. Remember, the tanks are having carbon dioxide. So you could have pumped carbon dioxide from what? From the tanks into the greenhouse. That will increase what? The amount of carbon dioxide inside the greenhouse. Or he could have added sodium bicarbonate to the soil. And why so? Because sodium bicarbonate in the soil will increase the amount of what? Or the concentration of what? Of carbon dioxide. Or the investigator or the farmer could have added the organic material that will decompose. We know that when organic material decompose, what do they release? They release carbon dioxide. So these are the other forms, or these are the other ways of what? Of enriching carbon dioxide into the greenhouse so that the amount or the level of carbon dioxide could increase. Now the second question says, explain how increasing the carbon dioxide levels to 0.1 can lead to an increased production of tomato. Remember here they said the farmer has increased the carbon dioxide from 0.03 to 0.1. Now the question says, explain how increasing carbon dioxide levels up to 0.1 can lead to an increased production of tomato. Don't forget the factors that are increasing the rate of what? Of photosynthesis. One of the factors that increases the rate of photosynthesis is the what? Is the increase in the carbon dioxide levels. So the answer here, you are going to base your answer looking into the amount or the concentration or the rate of photosynthesis. If the levels of carbon dioxide increase, what does that mean? It means that the rate of photosynthesis will also increase. And if the rate of photosynthesis increases, what does that mean? It means that there will be more sugars or there will be more glucose that will be produced. And if there's more glucose that is produced, it means that there will be more starch that will be stored, and then that starch will be used for what? For the growth of what? Of the tomatoes. So that is how the increase in the carbon dioxide level is so important. And then the third question, still based on the phrase above, they want us to name the 2 other factors which might have to be increased. Name other 2 factors that which might have to be increased to obtain a very greater yield when carbon dioxide levels, or when carbon dioxide level is increasing into 0.1. In other words, here you need to provide now other factors that can improve that can increase the greater yield of what? Of tomato, except for what? For the increase in the carbon dioxide. Here you are going to talk about other factors that increase the rate of photosynthesis, except for what? For the one that is already provided to us. Don't forget that that will be your light intensity as well as temperature. So that is how you are going to answer that question. And number 4, predict what would happen if carbon dioxide concentration within the greenhouse is increased to up to 0.5% for between 10 to 15 days if all other factors are kept constant. So predict. You must predict what is going to happen if the concentration of carbon dioxide increases up to 0.5. If it increases up to 0.5 between 10 to 15 days, but at the same time, other factors are being kept constant. So what is going to happen? Predict. Now, what is going to happen? There will be an increase in the rate of photosynthesis for that for 10 to 15 days. But after that, the rate of photosynthesis is going to do what? Is going to decrease. Why is it decreasing? It is because other factors are being limited. Other factors which contribute to the rate of photosynthesis are being limited. Factors such as what? Factors such as temperature and light intensity. If they are not being increased, what does that mean? The rate of photosynthesis will only increase for 10 to 15 days. After that, if it keeps on increasing, what is going to happen? It means that the rate of photosynthesis is going to decrease. Why is it decreasing? Because other factors are being kept constant. They were not increased. So that is our prediction. 5.2, here we are given the graph which shows the light intensity in arbitrary units, and then with the rate of what? The rate of or the mean of mass of lettuce plants after 21 days per grams. So let us quickly read into the provided statement. Scientists set up an apparatus to investigate the effect of temperature, the effect of light intensity, and the effect of carbon dioxide concentrations on plant growth. Using apparatus, they could control each other. The scientists set different temperatures, set different carbon dioxide, different carbon dioxide concentration. They also set different light intensity for 4 groups of lettuce. So they said these are the 4 groups of lettuce. Now, the average mass of lettuce plants serves as an indication of the rate of photosynthesis. In other words, this serves as what? As an indication of what? Of the rate of photosynthesis. Now let us look into the question. What is the influence of light intensity on the average mass of lettuce plants? What is the influence of light intensity? Remember here we were given different light intensity. So the light intensity here, this is our temperature. It starts with 15 degrees, and then we can see the rate of photosynthesis, or the average mass of what? Of lettuce plants. And then when light intensity is at 25 degrees, it has increased slowly a little bit. And then when light intensity is at 15 and the concentration of carbon dioxide is at 4%, it has now increased. And when the light intensity, or the influence of light intensity, increased to 25 degrees Celsius, which is the optimum light intensity for the rate of photosynthesis to take place or to increase. And then the carbon dioxide levels there were at 4%. Now the question says, what is the influence of light intensity on the average mass? What is the influence? You can see now when the light intensity increases, what happens to the average mass of what? Of the lettuce plants. They are also increasing. Can you see? And don't forget the questions, the statement said that the mass of lettuce plant serves as what? As rate of what? As rate of photosynthesis. Now let us check our answer now. The rate of photosynthesis increases, or the average mass of plants of lettuce plants increases as light intensity increases. So that is how you are supposed to answer that question, that when the light intensity increases, what is happening with the mass lettuce plant? It also increases. And don't forget that the mass lettuce plant indicates the rate of photosynthesis. That's why our answer will be in the form of what? Of the rate of photosynthesis, because the rate of photosynthesis is indicated by the mass of the lettuce plants. So that is how you are going to get your 3 marks. Name 2 limiting factors that influence the rate of photosynthesis at as the light intensity increases. Apart from the light intensity, what other factors will limit the rate of photosynthesis? So the other factors will be your carbon dioxide as well as temperature. Why are we now not including light intensity? It is because already light intensity appears from the question. And remember that there are 3 factors that are influencing the rate of photosynthesis. Now the question is saying, apart from the light intensity, what other factors can influence the rate of photosynthesis? That will be your carbon dioxide as well as your temperature. That was it about photosynthesis. And then the next topic, grade 11, is excretion. Now with excretion, the first thing that you need to know is your urinary system. You must be in a good position to, even if you are not drawing it, but you must be able to know the urinary system, all the parts which are involved with the urinary system. You must know the structure, the internal structure of the kidney. You must be in position to label all the internal parts of the kidney. You must also know the structure of the nephron. Be in a position to label all the parts of the nephron when it comes to what? When it comes to excretion. And then the last thing that you must know under this topic is homeostasis. And then the homeostasis control of what? The homeostasis control of water, the homeostasis control of water, as well as the homeostasis control of what? Salt, as well as the pH. Because all these homeostatic control are being catered or are being done in the what? In this topic of excretion. Thank you. Now, as I've said earlier on, that you need to know your urinary system as you know yourself. You need to know your the structure of the internal kidney as you know yourself. You also need to know the structure of what? Of the nephron by heart. Good. Now the first question says, draw supply or label parts A to G. In other words, you need to provide A, B up to G. So that is very, very, very simple, grade 11s. Only if you are prepared and only if you have read and studied the excretion topic. Now we know that on top or above each kidney there's a gland, and there are there's a pair of kidney, or there are two kidneys. On each kidney there's a gland there, and then the name of the gland is the adrenal gland. So let us quickly check the answers or the labels for the asked parts, or for the asked parts. A is our adrenal gland. Remember, is that gland that is found or is positioned above each kidney. So that gland is called adrenal gland. And then B is very simple. That is the kidney. And then what is C? Here is C. C is the the bladder. That is C. And then what is D? Here is D. D is the urethra. This is D, and then D is the urethra. And then what is E? We are trying to be in alphabetical order. What is E? Remember here, E and F are the vessels which supply blood in and out of the kidneys. So you must know which one takes in the blood into the kidney and which one takes the blood out of the kidney. Now, what is E? The E is this is E, the red one. E is the renal artery. And then F is the renal vein. And then the last part is G. What is G? That is the tubule that is coming from the kidney and then into the bladder. And then that tubule is called ureter. Ureter. You see, urethra without an R. So you must not confuse urethra with urethra. Urethra is the one that is taking urine to the exterior, and then in males it also takes that's where semen is also running through. And then ureter, it takes urine from the kidneys into the what? Into the bladder. That's where urine will be stored in the bladder temporarily. So without an R, ureter, without an R somewhere there, that is the one that is coming from the kidney into the bladder. And then urethra with an R there. That's how we are distinguishing between urethra and urethra. So the one with an R is the one that is from the bladder carrying urine to the exterior. So that is how you can distinguish, or you can differentiate between urethra and ureter. And then what type of blood does E bring to the kidney? What type of blood does E bring to the kidney? That is the deoxygenated blood. In other words, E brings unfiltered blood into the kidney. So that is the type of blood that is brought by what? By E into the kidney. What is the function of part labeled C? What is C? Already we have labeled all the parts. And then what is C? We said C is the what? Is the bladder. And then what is the function of bladder? We all know that bladder stores urine. Remember, I've just spoken about ureter that are coming from each kidney, and then they are conveying urine from the kidney. In other words, the ureter conveys urine from the kidney, and then it takes it into the bladder. So in the bladder, that's where this urine will be stored. So the question says, before I get there, what is the function of, what is the function of C? The function of C is to store urine. That is correct. Yes, is to store urine. And then name 4 important functions of part labeled B. What is B? We said B is the kidney. What are the 4 functions of the kidney? What does a kidney do in a human body? We know that I've said earlier on that it regulates water, it regulates salt, it regulates the pH. So the kidney is responsible for excretion, or it is responsible for the removal of what? Of waste. It is also responsible for the regulation of water. In other words, it is responsible for osmoregulation in the blood or in the body fluids. It is responsible for the regulation, for the balancing. When we talk of regulation, we are simply talking about the balancing. In other words, it is the kidney that balances the salt levels in the what? In the blood or in the body fluids. It is also responsible for the regulation of what? Of pH in the blood. So the the main functions is that it is responsible for the removal of all the waste, and then it is responsible for the regulation of water, for the regulation of salt levels in the blood or in the body fluids, and it is also responsible for the regulation of what? Of the pH. Which two substances does part D carry in adult males? So what is part D? So here is our part D. This is the part D. What is part D? We have labeled part D as what? As urethra, not ureter. Ureter is this one, but they want to know that in males, which two substances does urethra carry in males? Do not be too excited and set your own question. Read the whole question. It says, which two substances does part D carry in adult males? In adult males. So the first substance, we know that it carries urine, and then the other one, in adult males, it carries what? Semen. So urethra, that's where urine as well as semen runs through. So the answer there is urine and semen for 2 marks. The second part under the excretion, or under the topic of excretion, I've highlighted earlier on that you need to know the structure of the nephron as well. Now, with this drawing, or with this structure, you will never ever be expected to draw it. However, you will be expected to provide the labels of some of the parts. Now, before we can provide our own labels, I want to distinguish or to differentiate between the two blood vessels here. Can you see this arrow? This arrow shows this blood vessel, and then this blood vessel is the one that is getting inside the what? The Bowman's capsule. It is the one that is getting inside the Bowman's capsule. In other words, it is adding. So this is our afferent with an A. It's an afferent arteriole. In other words, it adds, it gets into, it is adding into what? Into the Bowman's capsule. And then as soon as it gets into the Bowman's capsule, what is happening? It starts now to branch, and then when it branches, it forms these blood capillaries, which are called the glomerulus. Don't forget that when it gets inside, then it is adding into the. That's why we are saying it is an afferent arteriole. And then and then it branches to form the blood capillaries, which are called the glomerulus. And then these blood capillaries, they reunite again. When they reunite, they now leave the Bowman's capsule, as you can see. When they reunite to form what now? To form the efferent arteriole. So the afferent is adding into, and then the E with an E, it is exiting, it gets out. So efferent, it shows that it is getting out, and then as it gets out, it forms now the efferent arteriole. So please do not be confused with the afferent and the efferent, and then you are now confused which one is afferent now and which one is efferent. know that the one that is getting into it is an afferent. How do you know that is afferent? It is adding. It's the one that it starts with an A. And then as soon as it gets inside, it forms the blood capillaries. It branches to form these capillaries. And then those capillaries are called the glomerulus. And then the glomerulus, they reunite again to form what now? To form the efferent. The E is leaving, as you can see the arrow. It is the vessel that is leaving the Bowman's capsule. And then that is the efferent exiting. And then as it goes now into the second part, so this is our first part of the nephron is the Bowman's capsule. And then the second part will be the the renal tubules. So the renal tubules, these are the renal tubules. As the efferent gets out, and then it now forms, it also reunites, it forms the blood capillaries, and then those blood capillaries will start to do what? To surround themselves into the renal tubules of the nephron. Now the first tubule of the nephron is the proximal convoluted tubule. So remember that the first one, the one which is towards the upper part, the one which is a little bit on the upper part is the proximal convoluted tubule. And then it goes down now to form the second part of the tubule. So this is the second part of the tubule. The second part of the tubule is the loop of Henle. And then don't forget that the loop of Henle is divided into three parts. We've got the descending, anything that goes down, we are saying it descends. We've got the descending loop of Henle, and then the bending part whereby it forms a hairpin structure. You can see it is the bending part of loop of Henle. And then the last part of loop of Henle, it's the ascending. Anything that goes up, we are saying it is ascends. So we've got the ascending loop of Henle. So it goes up to there, and then that is the last part of loop of Henle. And then a loop of Henle leads now into the last tubule. The last tubule is towards the end or is in the lower parts. And then the last tubule is the distal convoluted tubule. So do not be confused with the distal as well as the proximal. Proximal is towards the upper part, and then the distal convoluted tubule is towards the lower part. And then the distal convoluted tubule leads into the last part, which is the collecting duct. You see, it is very, very simple for you to do what? To label or to provide all these labels. So the question said, label the following nephron. So the answer here, I've already labeled it, but the clean answer is here. Don't forget the afferent arteriole getting in, the efferent arteriole getting out, and then the surrounding, the efferent will start to branch to form the blood capillaries, and then those blood capillaries are the peritubular capillaries. And then we'll be having the Bowman's capsule. This is the Bowman's capsule, and then what is inside the Bowman's capsule as the glomerulus. And then it goes now down to the first part, which is the proximal convoluted tubule, followed by the second part, which is the loop of Henle. I've already explained all the parts which are found in the loop of Henle. There's descending, there's ascending loop of Henle, and then loop of Henle will lead to the distal convoluted tubule, and the distal convoluted tubule leads to the last part, which is the collecting duct. And then activity 3, here we are given the structure of a nephron. Already you know your nephron by heart. You will be able to answer the questions which are now based on that. So the first question says, name the process shown at 1, at 2, at 3, and at 4. What are the processes which are shown there? Now, 1 is taking place, the process that is taking place inside the what? Inside the Bowman's capsule. And what is inside the Bowman's capsule? Don't forget that there are glomerulus. So it means that inside the Bowman's capsule, there will be that glomerular filtration. There will be ultrafiltration. So that is the process that is taking place at 1. And then at 2, what is happening? At at 2, there is reabsorption of whatever the substances which were able to pass through the podocytes or the Bowman's capsule into the proximal convoluted tubule. They will be reabsorbed by by the peritubular capillaries. So it means that there's reabsorption there. And then again, on the very same peritubular capillaries, there will be secretions of there will be secretion of sodium and all other substances which are being secreted there. So the process there is secretion. And then the last one that takes all the unwanted waste, that will be excretion. So you have already named all the processes which took place in 1, 2, 3, and 4. Let us check if our answers are correct. At 1, there's glomerular ultrafiltration. And then number 2, this is number 2. What is happening at number 2? There's reabsorption of other substances. And then what is happening at 3? There is secretion of some of the substances. And then what is happening at 4? There is excretion of all the unwanted waste. Mention 3 substances that move back into the blood at phase 2. What other substances? The substances which were filtrated and then they moved back at number 2. In other words, the substances which were reabsorbed. Remember, we said at number 2 there was reabsorption process there. So mention any other. Mention 3 substances were reabsorbed at 2. So the substances which were reabsorbed at 2 is your glucose, the amino acids, the vitamins, as well as water, because these substances are very, very important and they are needed by our bodies. Another question which is based now on the first part of a nephron. This is our Bowman's capsule. Now the questions will be based on the Bowman's capsule. You should know the parts of the Bowman's capsule. So in which region of the kidney will you find this structure? Where in the kidney does remember, in the kidney, let me quickly draw the structure of a kidney somewhere here in the kidney. And then there are 2 parts there. There's cortex, there's also medulla. So the cortex is somewhere there, and then the medulla is somewhere there. So in which part of the kidney will you find the Bowman's capsule? So definitely the Bowman's capsule, because it's the first part of the is the first part of the first part of the nephron. It is always found on the outer, but in the inner part of the of the kidney. So the outer of the inner is called the cortex. It means that it is found in the cortex of the kidney. And then the second question says, name the process in urine, name the process in urine formation that occurs in this structure. So the process of the formation of the urine that occurs here, what is happening in this structure? That is only ultrafiltration. So in other words, the ultrafiltration forms urine, because all the big particles, all the big substances will remain up there, and then the small substances which are not needed will pass through. So the formation of urine is done by by ultrafiltration, or by the glomerular filtration. Describe 2 structural adaptations of part C. Here is part C. Remember, part C is that the glomerular, the blood capillaries, which are called the glomerula. And now they want us to describe the 2 structural adaptations of part C for the process that happens there. What is the process that is happening there? The process that is happening here is ultrafiltration. Now, how is this glomerular, or how are these blood capillaries adapted to perform the filtration process? How are they made? How are they so that they are able to perform this process? Remember, the process that is taking place then is the ultrafiltration. So in other words, you must talk about the manner in which these blood capillaries are made, and for them to be able to do what? For them to be able to do what? To perform the ultrafiltration. You must also talk about the manner in which this glomerula is made for it to be able to perform the filtration. Now, the first answer, it says that the walls of the glomerula are thin, or they are single layered. In other words, if they are thin, what does that mean? It means that they allow diffusion to do what? To take place. For the process of diffusion to take place, it needs a thin or a permeable layer so that substances can go into and can leave the walls. And then the second thing is that it is made up of, let me clean here, it is made up of many podocytes. These are the podocytes. It is made up of many podocytes, or it is made up of the thin pores. These thin pores are called the podocytes. And then what is the function of the thin pores? What is the function of the podocytes? It allows filtration. In other words, it acts as a what? As a micro filter. It restricts large particles from passing through. Because they are so small, it means that the big particles cannot be able to pass through, but it only allows small particles to pass through. And then it is also made up of what? Of lots of what? Of blood capillaries. It's made up of lots of blood capillaries, and lots of blood capillaries, they ensure large surface area for the absorption of whatever that needs to be absorbed there. Yes, now we move to the third part of excretion, homeostasis. Remember, we said there's homeostatic control of water in excretion. There's homeostatic control of water. Let me quickly write it. There's homeostatic control of water. There's homeostatic control of salt. There's also homeostatic control of what? Of pH in the excretion. Now, let us look into the equations which are based on the homeostasis now. And before I can start with this question, with homeostasis, you must know the parts, you must know the part that secretes at length, which hormone does it secrete, and which hormone is responsible for the balance of water, which hormone is responsible for the balance of what of salt, and which parts secrete these hormones, and what the hormones, which organs are they going to target. Remember, that is what you need to know when it comes to homeostasis. Now, the first question here on the homeostasis is about the column. Remember, with the column, you state whether the option on B is A only, that is applicable on the statement at A, is B only, both A and B, or none. Now, the first question says, the blood leaving the kidney contains more of this substance than the blood entering the kidney. Is the blood leaving the kidney contain more of amino acid, contain more of carbon dioxide? What is the correct answer there? More than the blood that is entering the kidney. So the answer is it contains more of carbon dioxide. That is B only. Osmoregulation. Here we are given two hormones. Which hormone is responsible for osmoregulation here? Is it ADH? Is it a thyroid stimulating hormone? Or is it both the hormones, or none of the two hormones? So as I have highlighted earlier on that you must know the hormones responsible for the balancing of water and for the balancing of salt. So the hormone that is responsible to balance water is nothing else but antidiuretic hormone, which is ADH. So the answer is A only. Do not forget that. The tube that carries urine from the kidney to the bladder, which one? The tube that carries urine from the kidney to the bladder. This is the tube that carries urine from the kidney to the bladder. Is it ureter? Is it urethra? Or is it both of them or none of the two? The one that carries urine from the bladder to them, from the kidney to the bladder, it is ureter without an R. It is ureter without an R. So the answer is A only. The hormone or hormone secreted by the adrenal gland to regulate salt concentration of the blood. Is it ADH? No, we said ADH is responsible for water, so it's not ADH. Is it aldosterone? Yes, it is aldosterone. So the answer, the hormone that is responsible for the regulation of salt is nothing else but aldosterone. So that is the answer. Only one multiple choice question under homeostasis. If a drop in pH of the blood occurs, the kidney will A, increase the absorption of urine, or the kidney B, will decrease the absorption of sodium ions, or C, the kidney will decrease the secretion of hydrogen ions, D, the kidney will increase the reabsorption of bicarbonate ions. So the answer then is very simple. That is D. The kidneys will increase the reabsorption of bicarbonate ions. Activity 4. Remember, we are on the last part of excretion. We are now dealing with homeostasis. Now, activity 4, here we are given the statement and the diagram below. The diagram below represents one part of the negative feedback response that occurs when a person is dehydrated. We know that when a person is dehydrated, it means that the level of water in that person's blood is low. So that is an imbalance. Now the statement continues by saying the decrease in blood volume as a result of excessive loss of water is detected by what? By the brain. Now the questions are based on what? On the diagram as well as the statement. The first question says, identify a hormone X. What is hormone X? Grade 11s, not long ago, I think about a minute ago, I said when it comes to a homeostatic control, or when it comes to homeostasis, what you need to do, or what you need to know, is the hormone and what secretes that hormone. It can be a gland or the part in the body that secretes that hormone. And then you must also know the target organ whereby the hormone will send the impulse into, so that there will be response. Now the question here says, identify hormone X. Identify hormone X. Hormone X, what hormone are you going to talk about here in a regulation of what? Of water. In other words, you need to know the hormone that is responsible for the regulation of water, because water here is not balanced. So the hormone there will be your what? Will be your ADH. We said ADH, antidiuretic hormone. You do not have to memorize that big word, the antidiuretic hormone, as long as you know that ADH stands for antidiuretic hormone. That is the hormone that is responsible for the balance of what? Of water. Identify gland that secretes X. what gland secretes hormone X? What gland secretes antidiuretic hormone? Can you see here? It is the illustration or the diagram shows us that this gland is the gland that is situated at the posterior lobe of what? Of the brain. And what gland is that? And that gland is the one that secretes hormone X, which is ADH. What is the name of that gland? Nothing else but pituitary gland, because pituitary gland is the gland that is situated on the posterior lobe of what? Of the brain. So the answer there will be your pituitary gland. And then what is the target organ? Now, this hormone is going to send an impulse into this target organ. And then which organ is responsible for the regulation of water? When we were asked earlier on about the functions of the kidney, one of the functions is that kidney regulates water levels in the blood. So the target organ in these regards, it will be what? It will be the kidney or the kidneys. Why are we saying kidneys? Because humans have a pair of kidney. So in other words, we are all having two kidneys. So the target organs, there will be the kidneys. That's why that the answer is the kidney. Describe the response that occurs when a person is dehydrated. When a person is dehydrated, what does that mean? It means that blood has the low levels of what? Of water. That is an imbalance, meaning that homeostasis must take control here. Homeostasis must come in. Why homeostasis? So that the level of water can be increased. But the question says describe for about 5 marks. When you describe the response, in other words, you're going to talk about the whole process of doing what? Of increasing the amount of water in the blood. So how do you answer that? The first one is that ADH will cause the wall. The ADH will cause the walls of the kidney tubules. Remember the kidney tubules, those renal tubules, your collecting ducts as well as your distal convoluted tubule to be what? To be more permeable. Why ADH? Because ADH, the more the ADH in the blood, the more the permeability of what? Of the walls of what? Of the kidney tubules. And then when the walls of the kidney tubules become permeable, what does that mean? It means that more water will leave what? The more water will leave the distal convoluted, the kidney tubules, your distal convoluted tubule, as well as your collecting duct. And then where will that water go to? The water will leave because of the permeability of the walls. It will get now into what? Into the blood vessels, into the blood capillaries. And then if it gets into the blood capillaries, what does that mean? It means that the volume of water in the blood will increase. And what does that mean? Again, it means that urine will be less because more water has been absorbed by what? Has been reabsorbed by what? By the blood capillaries. And then it means that the water or the amount of urine that is going to be produced will be less or will be little. And that little urine that is produced will be so concentrated. In other words, it will have little water in it. Why little water in it? Because more water has been reabsorbed by the blood capillaries. As a result, the level of water in the blood will increase back to normal. So that is how we are supposed to describe the answer for this question. And then on this one, I will give you, let's say, a minute to do it. But before I can give you a minute to do it, let me quickly go through with you, together with you on the question and on the statement. Through homeostasis, the kidneys control the composition and the volume of blood and tissue fluid. The table below compares the concentration of different substances in blood. It compares the different substances in blood. Yes, it compares the different substances in blood. Yes, and then in the you need to study your the table that is provided, and then to answer all the questions which are based on this table and the story above the table. So I am giving you only a minute to answer this question. Okay, grade 11s, we are back. We are back. I want you to compare your answers or your response with my answers. The first question says, state what is meant by homeostasis. That is very simple. What is homeostasis? So homeostasis is the maintenance of a constant internal environment. I'm sure you got the very same answer. Question 2, explain the difference in concentration of a glucose in the filtrate and in the urine. Now on this one, you are going to concentrate on the glucose as a substance because the question is about it. And then you now explain the difference in the concentration in their filtrate, filtrate as well as the urine. So you need to explain the difference in the concentration of glucose. You are going to focus on this one, and that one. What is the difference there? So I want you to check your answer versus my answer, to compare your answer with my answer. Glucose is completely reabsorbed. As you can see, glucose in blood, it was 0.1%, and then on the filtration is, remember that the filtration is taking place in the glomerulus there, meaning that the whole 0.1 percent was there. And then in the urine, what is happening there? There's nothing, meaning that the urine does not have what? Does not have glucose. Now the answer is that glucose is completely reabsorbed in the proximal convoluted tubule by an active absorption or against a concentration gradient and pass into the blood capillary. So it went back to the blood capillary, meaning that the 0.1 which was filtrated, it was reabsorbed, went back to the what? To the blood. So that is the difference. As a result, our urine does not have glucose in it, or the urine does not have glucose in it. And then explain the difference in the concentration of ammonium ions in the blood and in the urine. So here from the table, we are going to look into the ammonium ions. And then what are you going to look into? You are going to check the blood substance, as well as in the urine. So you are going to concentrate on the three. You check the substance here is the ammonium ions, and then you are looking into what? You are looking into the blood as well as the urine. So you need to differentiate between the ammonium ions in the blood and in the urine. So check your answer with my answer. What is happening? Ammonium ions are excreted. Ammonium ions, where are they? They are excreted in the urine, meaning that they are going out as a form of, in the form of urine. So they are excreted from the epithelial cell of the distal convoluted tubule, and then into what? Into the tubular fluid. That's why they are excreted. The ammonium ions are excreted there. It means that the blood does not have the ammonium ions. That is the difference between the two. State the percentage of glucose reabsorbed in the nephron based on the results shown in the table. In the blood, glucose was 0.1. The concentration of glucose, or the amount of glucose, was 0.1. And then what was reabsorbed, it was filtrated and reabsorbed. The whole amount of 0.1 was also 0.1 was in the blood, and 0.1 was reabsorbed. So what is the percentage of the reabsorption of these? It is 100%. 0.1 was in the blood, and 0.1 was filtrated, and 0.1 was then reabsorbed. It means that the whole amount of glucose was reabsorbed. So that is 100% in the form of percentage. Through homeostasis, or is the very same story that we have read. Now the question says, describe the role of ADH when water content of blood is low. When water content of blood is low, in other words, when there's little amount or low levels of water in the blood, describe the role of ADH. What does ADH do when the level of water is low in the blood? So the answer is very simple, that the hypothalamus in the brain is stimulated, and then as it is stimulated, it will send the impulse into the pituitary gland. Remember, we are not using a word message for the impulse. It sends the impulse. It does not send a message. We know that an impulse is a message, but scientifically speaking, they are sending an impulse, not a message. So after the hypothalamus has been stimulated, meaning that the hypothalamus of the brain is the one that is detecting the change, then that's how it gets stimulated. Therefore, it will send the impulse where? Into the responsible gland. What is the responsible gland there? Is the pituitary gland. Why pituitary gland? Because pituitary gland is the gland that is responsible for the secretion of what? Of ADH. So it means that pituitary gland will secrete more ADH. Why ADH, not any other hormone? It is because ADH is the hormone that is responsible for the regulation of what? Of water. So that's why pituitary gland secretes more ADH. And why more ADH? So that the walls of the distal convoluted tubule, or the walls of the renal tubules, or the walls of the walls of the kidney tubules are becoming more permeable. So you see, it secretes more ADH so that the walls of the kidneys become more permeable. So these are very important words. More ADH is secreted to secrete more, is secreted so that the walls of the renal tubules become more permeable. And then if they are more permeable, what does that mean? They will be more permeable. As a result, the sodium will be pumped, will pump the sodium ions into the medulla, and then the gradient is set up for what? For osmosis. Remember, osmosis is the movement of water from the higher water potential into the lower water potential. So that water leaves the tubules by osmosis. Water will leave from the higher water potential into a low water potential and enters the medulla, and then water enters the bloodstream. The water will enter the bloodstream, and then as a result, the amount of water in the bloodstream will do what? Will increase back into normal. So you see, the important things here that you must always know is that the pituitary gland will secrete ADH. In this instance, it will secrete more ADH, so that more ADH makes the renal tubules to be more permeable. That is it, grade 11s. And then this one was taken from a DPE 2018. This was a metric paper. Why metric paper? Don't forget that homeostasis is also done as is in grade 12, meaning that if you master it now and then when you get to grade 12, you are not going to struggle. So this question basically was taken from the DBE May-June 2018 paper. Now, an investigation was carried out to determine the influence of alcohol on the volume of urine produced. Now, 12 healthy 23-year-old males of similar height and similar mass participated in the investigation. The investigation was conducted as follows. Men were divided into 2 groups of 6 each, and then group A as well as group B. The 2 groups ate the same food, and then they did the same exercises, or they did the same exercise for 24-hour period before testing. Now, each group was given the following to drink after 24-hour period. Group A was given 1 liter of alcohol-free, in other words, non-alcohol beer. And then group B was given 1 liter of alcoholic beer, the one that contains alcohol. Now, urine was collected from each man every hour. The urine was collected from each man every hour. Assume that the volume of urine collected is equal to the volume of urine produced. Now, the results were reorganized in the following table, and then time versus average of urine collected in milliliter. Time 1 hour, after 2 hours, and after 3 hours. And then urine was collected from group A men, and it was also collected from group B men. Now remember, this is an investigation question, and then when you are answering or when you are dealing with the investigative questions, what you need to do to know, first thing is that you must be able to identify, or you must be able to identify the aim, the independent variables as well as the independent variables. And then the aim is already stipulated out on top there. And then again, you must be able to identify or to check the aspects or the factors which were taken into consideration, making the investigation to be reliable. Which aspects were followed to make sure that the investigation was reliable? In other words, you check the reliability of the investigation. And then you must also look into the aspects which made the investigation to be valid. You are now looking into the validity of the investigation before attempting the questions. And then you also check your control versus experiment, all the apparatus for the control and the experiment. You need to know the variables which were kept constant, all your controlled variables. They are there, and then if they are not enough, you must also think of other variables which are not maybe mentioned in the investigation that could have kept constant or which could have been controlled. Now the first question says, state the dependent variable in this investigation. Let me go back a little bit. The dependent variable, remember, is the variable that is measured. We did this in our previous lesson, that when we talk of the dependent variable, we are simply talking about the variable that is being measured. So what is being measured here? What is being measured here is the volume of urine. So the dependent variable will be what? The volume of urine. State the 2 planning steps the investigators had to take before the investigation could start. So we also talked of the planning steps when you are going to do your investigation. We did that in our previous lesson. So the investigator should have decided on the time, on the date, on the place to conduct the investigation. That is the first thing that he was supposed to do or to decide on. And then he could have he was supposed to decide on the apparatus to be used, all the materials that are needed for the particular experiment that he was going to use. And he must also decide on how to collect and record the data. And then he was also supposed to advertise for the volunteers so that all participants were there voluntarily, so they were not forced. And then he must also decide on the number of participants to include. Here we had 6 males and 6 males, so that's why he decided on that number. He also decided on the factors which were kept constant, which were kept controlled. For an example, factors to be kept constant. So he also had to decide on that. So these are all the planning steps. Now, state 2 factors to remain constant other than ones that already mentioned. So the factors that could have remained constant: same room. Remember, when talk of the controlled or the constant variables, you must not forget a word same. The same room, the same temperature, the same environment, the same apparatus to be used, the same investigator. They must not be investigator A, and then for the second readings, there's different investigator. There must be the same investigator. There must be no any other liquid that will be taken by both groups, group A and group B, and then they must all do the same activity. So these are other factors that would have remained constant during the investigation. The diagram below shows the reabsorption of salt and water through the tubules of a nephron in the kidney under the 3 different conditions. 1, 2, 3 conditions. The width of the arrows represents the amount of salt and water. So here is the key. The black one represents the salt. So this is the salt. All the black arrows represent the salt, and then all the light gray arrows represent water. So these are the arrows of water. Now, question 1 says, name the hormone in the human body that is responsible for controlling the content of water, or water content. The hormone that is responsible for water is ADH. We all know that. The one that controls salt is aldosterone. Name that gland that secretes the hormone in question 1b. That gland that secretes aldosterone, that is the adrenal gland, which is found on top of each kidney, is the one that secretes that. Which diagram, 1, 2, or 3, would represent a person who had eaten salty chips on a hot day without any intake of water? So how are you going to answer that? How are you going to distinguish between 1, 2, and 3, that the person has eaten salty chips? If he has eaten salty chips, it means that the level of salt in blood will be high, and then the amount of water or the level of water will be low, because they said on a hot day. Because on a hot day, what is happening? We always sweat, and then when we sweat, we lose water. So out of these 3 people, what are you going to check? What is going to be reabsorbed here? Let us check reabsorption of salt. It means that this person needs salt, but here they say person ate a lot of salt, so this one is out already. And then second one, diagram 2, again, salt is reabsorbed, and there's no way that salt can be reabsorbed because already salt is high in this blood. And then here, salt, little salt, can you see, is reabsorbed. So I think diagram 3 represents that person. And then high amount of water. Remember we said gray represent what? Represent water. And then gray, there's also high amount of water that needs to be reabsorbed. Why? Because that person lost water. So diagram 3 is the answer. Describe the role of aldosterone when the water content and salt concentration of blood is low. So with this one, I'll just project all the answers as they are. More aldosterone is secreted by adrenal gland, because adrenal gland is the gland that secretes aldosterone. So more aldosterone will be secreted, and then the walls of the distal and the collecting duct will be more permeable. And then what does that mean? It means that more sodium will leave the kidney tubule and enter the medulla. So it's the same answer from the previous question, where it is absorbed into the bloodstream, or where it is reabsorbed into the bloodstream, and then the salt level in the blood will increase back to normal. And then the second last on the homeostasis, we are given the table and the story there, and then the question says, state with a reason which of the following parts A, B, C, or D of the nephron represents A, the afferent arteriole. Afferent, remember, afferent is the one that is getting into the Bowman's capsule. So by merely looking into the table, you can see that the flow rate at D is high, meaning that that's where there's high amount of blood. So afferent is afferent will be D. Why? And then they said with reason why, because it contains the proteins. There are proteins there, and then it is also having the highest flow rate. And then the one that represents Bowman's capsule, which one represents the Bowman's capsule? It's going to be B. Why? Because the concentration of glucose is high there. The concentration of glucose at B, waste glucose, is high, is at 0.10, and there is no protein which is found in the in the Bowman's capsule. The one that represents the loop of Henle, remember in the loop of Henle, when you think of loop of Henle, you must always think of what? We must always think of your sodium ions. So which one has the sodium ions there? It is C. You can see it is C, because there's no glucose at C and there's sodium ions. And then which one represents the collecting duct? And then the collecting duct obviously is the one whereby urea is highest. So that will be your A, because of the high concentration of urea. And then the last topic for today is cellular respiration. Let me not waste time. Let us provide the correct terminology for each of the provided statement. For each of the following statement. The process during which glucose is converted into pyruvic acid, it is glycolysis. And then the gas which is essential for the Krebs cycle to occur, the gas will be oxygen. The folded structure found on the inner membrane of mitochondrion, the folded, those are the crystalline. The stage of aerobic respiration that releases carbon dioxide, that will be the Krebs cycle. The genetic material found in the mitochondrial matrix, that is mitochondrial DNA. That is very simple. The stage during aerobic respiration when water is released as a waste product. What is that stage called? Oxidative phosphorylation. And then the last on the terminology, the organelle in which respiration takes place. That is very, very simple. That is our mitochondrion. Second question here is our the column. I want to give you only 30 seconds to do the this activity, the column. Only 30 seconds. and then we'll get back. 30 seconds. Okay, we are back now. Before we can do this activity, there's a question that says, ma'am, how do we distinguish between the renal artery and the renal vein in a black and white picture? Okay, that is a very, very good question. How do we distinguish between the two? Remember, we said renal artery, it is taking the deoxygenated blood into the what? Into the kidney, and then the renal vein is taking what? The oxygenated blood out of the kidney. In other words, if the diagram is not in a colorful way, it is in black and white, they will always use what? The arrows to show that this one is the one that is getting into the kidney, using the arrow, and then the one that is getting into the kidney, it is the renal artery, and then the one that is leaving the kidney, it is the renal vein. So there will always be arrows in a black and white diagram. A very, very excellent question. Thank you so much. Going back to the questions on question activity 1.3, end product of anaerobic respiration in humans, that will be the lactic acid. That is the end product. A product of cellular respiration which supplies energy needed for metabolic reactions in cells. That will be your ATP. The fuel required for cellular respiration, nothing, neither of the two. It is not glycogen, it is also not glucagon. Molecule that stores energy, that will be your ATP. And then you must also be in a good position to draw and fully label the structure of the organelle which is responsible for cellular respiration. And in this regards, the organelle will be a mitochondrion. So you must start first with your heading, the mitochondrion, a beautiful structure that is correctly drawn with the correct labels. Remember that here on the outer, we've got the outer membrane, and then with the inner membrane, and then inside the inner membrane, that's where we've got those folders which are called the cristae, and then inside the cristae, that's where we are having the matrix. Matrix is inside, and then inside the matrix there are ribosomes. So that is also a structure that you should be able to draw and fully label, because it's a simple drawing to do, whether in a test or in the examinations. So we have come to the conclusion of our lesson, grade 11s. But before we can say goodbye to each other, don't forget to take the following to the exam room. You must have your ruler, which has lines. You must have your protector. If you are asked maybe to draw the pie chart, at least you are safe. You are having your protector together with your compass. You must have 2 pens, so that if the first one stuck and then you are having an alternative pen, you don't start to borrow from other learners, because you'll be disturbing them. You must also bring pencil with an eraser. Don't forget your simple calculator. So with those words, grade 11s, we have come to the conclusion or to the end of our lesson. Until we meet next time, have a very good day. Bye-bye.